Now, good evening, colleagues, fellows, members, ladies and gentlemen. The title of this evening's webinar is Technology Shaping the Future of Surgery. Some of this webinar has had to be pre-recorded, so just bear with us. Professor Ronan Cahill is going to chair the webinar this evening, and Professor Cahill was appointed Professor of Surgery at University College Dublin and the Mater in June 2014 before taking over leadership of the section and subject of surgery in UCD in 2017. In 2019, he was appointed Director of the Centre for Precision Surgery, UCD, the Digital Surgery Unit at the Mater. There are three presenters. We have fluorescence-guided surgery, we have genomics and its future impact on surgery, and the future direction of robotic surgery. We'll be starting with genomics and its future impact on surgery with Dr. Simon Fernie. Dr. Simon Fernie is currently a senior lecturer in genomic medicine, and he has established the Genomic Oncology Research Group at RCSI. This will be followed by fluorescence-guided surgery, where we have Professor Donal O'Shea. Professor Donal O'Shea is head of the RCSI Department of Chemistry. His central theme of his research lies in the advancement of new strategies for the synthesis and functional assessment of structurally complex molecules. And he's also involved in the development of NIR, fluorescence-guided surgery. We then have Colin Pearce, who's going to present the future direction of robotic surgery in Ireland. And Mr. Pearce was appointed to the UL Hospital Group as a consultant colorectal and general surgery in 2016. Prior to this, he had done a fellowship in the Cleveland Clinic. And he has pioneered the robotics team who uses the Da Vinci dual console robot. So you're all very welcome. So I'd now like to hand over to Professor Ronan O'Cahill.
So, President, thank you very much for the introduction. And I'm really delighted to be chairing this particular session and webinar. I think it's really interesting to think about the new technologies that will define our practice now and in the near future. And we're really lucky to have such good surgical research happening in Ireland and in the College of Surgeons. So this is a little different to the normal session, which has been about state-of-the-art current practice. Here we're going to look at the possible, probable direction of surgery into the near future. And I'm delighted that there's such a strong genomics program developing in RCSI, and it's great now to hear Simon Fernie's insights and perspectives of how this actually applies to surgery and making care of the surgical patient better. So, Simon, it's great to have you. Please go ahead whenever you're ready.
Thank you very much, Ronan. I'm just going to share my presentation now. I hope you can all see that.
Yeah, I can see it very well. Can hear you very well.
That's great. And thank you for the introduction, President and Professor Cahill, and thank you for the invitation to talk to you this evening about genomics and its potential future impact on surgery. So just to introduce myself, I am head of the Genomic Oncology Research Group at the Royal College of Surgeons in Ireland. So my background is in bioinformatics, which is genome data analysis. So my group doesn't do any wet lab research. We are all computer based and we essentially crunch data. So just so you're aware of my background. Okay, so I'd like to touch on what I think is going to be happening over the next decade at this interface between research, genomics education, and genomics in the clinic and in the surgery. So there was an opinion piece in the Lancet actually about 3 years ago highlighting genomic medicine and giving a stance that to maximize the benefits of genomic medicine, it is crucial that research is interwoven with clinical implementation. So I'm going to give you a brief history of genomics, take you through what's possible with genomics, what's happening globally, and then what can be done in Ireland. So all of this is based on the Human Genome Project. So this was a global initiative which was set up in 1990. And the goal of this was to publish and sequence one human genome, and for this genome to be the reference human genome for all genomics and biomedicine research. So this was a very ambitious endeavor, and the first draft of this was published in 2001. So this took 11 years for the draft, and it was finally completed by the International Human Genome Sequencing Consortium, as it was known then, in 2003. So 20 years ago, basically. So this first human genome took about 13 years to complete, and at a cost of about $2.7 billion. So that's an astronomical amount of work and financial cost to do one genome. So where are we today, 20 years later? So actually, the future is now, and we've seen a genomics revolution take place, in particular over the last 15 years. So this graph is the cost of how much it costs to sequence one human genome. So back at the start of the millennium, this was still up in tens of millions of dollars. But you can see from the graph, there's a huge drop in sequencing costs around the year 2007. And this is because of a new development in technology known as next generation sequencing. So the Human Genome Project used a technology called Sanger sequencing to sequence the reference genome. So this next generation sequencing technology is based on the initial approach, but what it does, it sequences DNA in a massively parallel fashion. So this increases the amount of DNA that can be sequenced, and you see the reduction in cost associated with this technological development. So this is got better and better, and improvements have been made over the last 15 years. And we can see in the last 5 years, it's dropped down below $1,000 to sequence a human genome. In fact, we can do this commercially now for less than €500 if we outsource to a sequencing provider. So it's really in the research realm, and I just want to highlight here how fast-paced genomics is. So, in fact, these developments are coming so quick and fast. This is an announcement from last month where the main sequencing provider in the world, a company called Illumina based in California, has developed a new sequencer which is going to result in genomes being able to sequence at a cost of about $200 a genome. And as I highlight now for only the cost of a few dozen cheeseburgers, you could have a fully sequenced human genome. So perhaps if you were thinking of investing €200 or so in cheeseburgers, you might want to think again and have your genome sequenced instead. So I'm going to give you a brief introduction as to how we actually go about sequencing a human genome. So essentially, we're starting from a tissue sample from a patient, so that might be tumor tissue, or it might be a blood sample. We're extracting DNA or RNA, if you're sequencing RNA, from the cells in the sample. And what's happening then is we can't work with the long stretches of chromosomal DNA. So this is fragmented into shorter stretches of DNA. And then what we do is we attach specific adapters onto the end of these short DNA fragments. And this allows the DNA to stick to a glass slide, which is known as a flow cell, which is then inserted into a sequencing machine. This is an example of one here on the left and on the right. And then the sequencing chemistry sequences these short fragments of DNA and spits out the sequencing reads in nucleotide stretches. We then use computational algorithms to map these short stretches of DNA back to the reference genome that the Human Genome Project sequenced initially to work out the genome from the individual that you've sequenced. So that's it in a nutshell. So what are the applications? If we do a whole genome sequencing experiment, what results are we getting? So in this figure, we're seeing all the little short reads of DNA that I talked about that come out of the sequencing technology, and these are mapped back to our reference genome. So we can see from this individual here, most of the reads, the sequencing reads, are mapping back to chromosome 1 from the reference sequence. So we can compare the DNA sequence from the patient sample that we've sequenced back to the reference sequence. And if we see here on the left-hand side, we have an A, in the nucleotide A in the reference sequence, but we see the nucleotide C in the reads from the patient sample, indicating a point mutation in the genome of the patient that we sequenced. We can also pick up short insertions and deletions in the DNA. And also by mapping these reads back to the reference genome, just by seeing the number of reads that map back, we can identify copy number alterations. So regions of the patient's genome of the chromosome that may have been lost or gained or amplified. And this is obviously very relevant in terms of cancer, where we see a lot of gains and losses in tumor development. What we can also do is we can pick up structural variations. For instance, a chromosomal translocation. So this is where something's gone wrong during DNA replication, and one end of one part of a chromosome has been stitched to another chromosome. So we can see here on the right-hand side, we have a number of reads. In blue, we have the fragments of DNA. Half of the sequencing read is mapping to chromosome 1. The other half is mapping to chromosome 5. So this is indicating that a chromosomal translocation has occurred between these two chromosomes. As I said, this is common again in cancer, for example, the Philadelphia chromosome. Importantly, we can also map our sequencing reads from the patients to other genomes. So to non-human sequences, to microbial genomes, to viral genomes. And this is obviously very important, what with the increasing recognition that the microbiome plays in disease pathogenesis. So, as I said, I'm head of the Genomic Oncology Research Group, so most of my research is focused on cancer. So apologies if you're not in the cancer domain, but all of this is relevant to other disease areas as well. Specifically in cancer, though, we want to find out the mutations and alterations that have occurred during tumor development. So in order to do this, we need to take a sample from the tumor sample from the patient, and also a matched normal sample. So this is normally from the blood. So what we do is we sequence the somatic genome, the tumor genome, and also the germline genome. And then we map both of these to the human reference genome, and then we compare the tumor and normal reads to identify the somatic variants that have occurred during tumor development. And on top of this, we also have our germline genome, so we can have a look at cancer predisposition genes, and obviously of relevance to other disease areas. We can look at rare and common variation in that patient's genome that might be contributing to disease development in the patient. Okay, so I've focused here on genomics, but there are many applications of next generation sequencing, not just genomics, also transcriptomics and epigenomics. And when I'm talking about these different areas, when I'm talking about genomics, we're essentially looking at changes in the DNA or genome sequence. So as I said, this can be done by whole genome sequencing. It can also be done by exome sequencing, which is a technique which just sequences the coding regions of the DNA of the genome. So that's the 2% of the genome that actually results in proteins. We can also look at transcriptomics, so that's sequencing RNA to look for changes in gene expression. Epigenomics relates to changes in the regulation of gene expression, and there are a number of different assays that can be used for this. Also, in the last 10 years, there's been a huge drive in single-cell omics. So this is sequencing in particular the RNA, but also the DNA from individual cells. So we can use this to look at the heterogeneity between cells and cellular populations. And this is obviously very relevant in cancer with the inherent tumor heterogeneity you see. So we can do all these different types of assays, but we're using the same sequencing machines to sequence the reads and to spit out the sequencing reads that we analyze at the end of this. So that's an overview of what can be done with the sequencing technologies. What is being done? How is genomics being implemented into personalized medicine around the world? Well, this has exploded over the last decade with initiatives such as the Precision Medicine Initiative in the US, Significantly Genomics England, and the 100,000 genomes in the UK. This is happening in global initiatives, so ICGC, which sequences tumor genomes from a number of different countries worldwide. Individual countries have sequencing initiatives, and also there's the European 1 million genomes project, which is hoping to sequence more than 1 million genomes within Europe. Unfortunately, this is not happening at a national level in Ireland, but hopefully we'll start catching up soon. But in the meantime, we can look enviously across the water to the UK. We're building on the success of the 100,000 genomes project from Genomics England. The UK government launched Genome UK a couple of years ago. So this is to build an NHS genomic medicine service. And the goals of this project are to be the first national health service to offer whole genome sequencing as part of routine care. Okay, they're starting this off by sequencing childhood cancers and also rare disease. But the goal, as I said, is to extend this access to molecular diagnostics and to offer genomic testing routinely to all people with cancer, rare disease, and undiagnosed disease. And also looking forward to be able to link and correlate these genomic data to provide new treatments and diagnostic approaches to help and make informed decisions in patient care. So as I said, we're not there in Ireland at the moment. Hopefully that will change soon. But what can we do in the meantime? So I just want to give you a quick flavor of some of the genomic research projects we're doing in my group in collaboration with surgeons and with medical oncologists in Ireland. So this is along the lines of just single genomic case reports or larger cohort studies, or doing longitudinal monitoring of cancer patients using genomics. So an example of this is a project we did in collaboration with St. Vincent's University Hospital, where we're looking at synchronous colorectal tumors. So these are tumors that patients who have multiple tumors at diagnosis. So we looked at 3 patients who had between 2 and 5 synchronous tumors. And we did whole genome sequencing and RNA sequencing to see where these tumors related in each patient, or had they arisen independently. So from our whole genome sequencing results, we found in fact that these tumors seem to have arisen independently, and in that they had different mutational profiles in each of their tumor genomes. We also did another study using exome sequencing in locally advanced rectal cancer. These are patients who undergo neoadjuvant chemoradiation therapy. We took pre-treatment biopsies, on-treatment biopsies, and then surgical resection samples. And we did exome sequencing of all of these samples to compare tumor evolution through the course of treatments. Finally, we can implement this within clinical trials as well. This is a HER2-positive breast cancer clinical trial where we did exome sequencing of diagnostic biopsies and then pre- and post-treatment biopsies, and also monitored patient response using circulating tumor DNA. So these are just examples of what we can do now in Ireland. So in terms of genomics and its potential future impact on surgery, I think this is an open dialogue that we can start having now, but obviously it's going to be at the intersection of research, education, and in the clinic. Importantly, in terms of research, I believe there's a strong argument that the current and next generation of clinicians, surgeons need to be genomics literate, and that genomics and next generation sequencing should be part of the undergraduate and postgraduate curriculum. So this could take place through a form of lectures and practicals to develop a wider understanding of genomics data and the analysis of these types of data. In addition, a really nice model that we've used in my group over the last 10 years is joint supervision of research projects between a surgeon or a clinician and a bioinformatics principal investigator, where we can have joint supervision of a clinical research fellow undertaking an MD or a PhD and a data analyst on specific projects. This has worked really well for us, and we've had several collaborations and publications using this model over the last 10 years. Finally, specifically in cancer again, this is a longitudinal process where we're not just looking at one time point. We're taking a snapshot at different time points of the patient journey. So at diagnosis, during and after treatment, follow-up, relapse if the patient relapses, and further relapses. So genomics data can be used at all these different time points. You can take different samples at these time points and then compare genomic evolution through the patient journey. So we're actually doing this. We're trying to do quick whole genome sequencing of patients within the treatment window for a patient. We can actually get results back from a sequencing provider within 14 days of having sent the sample away. And my group analyzes the data, and we've been involved in some multidisciplinary team meetings to discuss the results of the whole genome sequencing analysis. So this is what's happening already in the UK and in the US, and it's something we can think about implementing already in Ireland. So finally, some further aspects. I've talked about this next generation sequencing technology where we use these short reads. The next iteration of this, called third generation sequencing or long read sequencing, is able to sequence longer reads, which makes the analysis easier to do, makes it more accurate in mapping the reads back to the reference genome. Single cell analysis is becoming more and more prevalent. Both long read and single cell analysis are more expensive to do. So they're really in the research domains at the moment, but they will eventually work their way into the clinical environment as well. Liquid biopsies, looking at circulating tumor DNA or tumor cells to look for a tumor development, and also longitudinal monitoring of patients is huge at the moment, is going to get bigger and bigger. A really important aspect of this, which I don't have time really to go into, is the GDPR aspects of this, in terms of genomic data are classified as sensitive data under the health research regulations and GDPR. So this is something we need to be cognizant of and manage this going forward. So I just like to thank you for your attention. I'm happy to take any questions. My details are here at the bottom. If you'd like to email me, please feel free to do so. I'm happy to talk about this or discuss any collaborations. And thanks again for your time.
So Simon, that's really awesome. That's a great overview of the current state of the art and the I guess the capabilities that's happening. So you kind of hinted at just at the very end, the idea of genetic exceptionalism. Where do you stand on that? Do you think that's this idea that it's special, and other people argue that it's just data? Where do you think the stand is? I know what GDPR says it is right now, but what's your own thoughts about where it should sit?
So my thoughts are, I guess a lot of people are happy to contribute their samples and then their data to medical research. And I think it's important, education is really important this, because it's important to understand that it's not just your DNA, it's really your family DNA. So you're sharing that with any siblings, your parents, relatives, and also your children. So if that data isn't protected properly, you might be giving away information that you don't necessarily want to be, in particular for your relatives and for your children. So we have to be really careful here and to go into it with our eyes open.
Yeah, okay. You gave your email there looking for people to reach out to you. How do you, I mean, there's so many things that genomics can be applied to. How do you triage what's a good project, or what should people, if they have an idea and they want to collaborate, what are the core things they need to bring to make a successful project with you?
So I think it's really the research question is obviously key. It's identifying an angle where you have a speciality that hasn't been addressed before. And obviously the best way to do this is to write a grant. I'm happy to sit down with people and discuss writing a grant, because that's how you triage really whether an idea is good, and hopefully you'll get the funding. Because look, it is expensive to run a lot of these projects, and you have to build in the genomics sequencing and the data analysis. But it's on the flip side of that also, there's an awful lot of data out there in the public domain, which we use. So if you have a specific question, there may already be data generated out there that we could use to answer a question. So come with a specific question, and then we can sort of see what can be done to answer that.
And who's funding this type of work now? Does this come under HRB, SFI, Wellcome Trust? Where do you think the most productive grant opportunity is? So
all of the above. I've been funded by SFI, Wellcome Trust, and HRB to do genomic sequencing studies. So it's as long as it's a nice idea, I don't think the cost or the fact that you're doing genome sequencing is a limitation to any of these funding bodies.
Okay, awesome. So there might be some questions coming through. We'd encourage anyone watching to raise some points, and we'll try and get back to them at the end. But Simon, that's been a terrific primer in the field. Thank you very much.
Thanks a lot.
We're going to talk next then about fluorescence guided surgery, which is both the real thing now and it's going to get more and more, I think, interesting and exciting. I'm going to present a little bit of the clinical work at the moment, and then I'm going to hand over to Professor Donal O'Shea, who's the head of chemistry at RCSI, and Donal has a first-class research lab that's building the fluorophores of the future. So I hope, Donal, I do you justice in this clinical intro, and we really look forward then to seeing you and hearing you. So first step is sharing my screen, and next step is to put it onto slideshow. I hope that's clearer and showing to everybody. Fluorescence guided surgery is using agents to try and give us an extra layer of information about the tissues. And it's pretty easy right now because there's only one of these agents, a dye called indocyanine green. It's rather biologically inert. It can be given intravenously. It's excreted or metabolized by the liver. But what's cool about it is that it fluoresces. It can absorb energy at one wavelength and re-emit it at another, and these are visible and can be seen. So there's a lot of clinically available systems that have cameras. All the major laparoscopic stack companies have a system, and it's really very interesting to see how seeing perfusion in real time is impacting clinical practice. So I'm a colorectal surgeon. We tend to give it routinely in our cases. And this is what you see. This is an extracorporealized right colon. You see the transverse colon and the small bowel still have their blood supply intact. There's a cancer over there near the ascending colon. The drug is given within a few seconds. It circulates, reaches the tissue, and you see these rather beautiful pictures of the perfusion status of the bowel. You see where I've divided the vessels with a high ligature. And this is giving us a sense of that dynamic perfusion of the bowel at the moment when we're going to make a decision about where we're going to transect and what bits are we going to join together again. So there's no augmented reality here. This is just reality being displayed via this multispectral analysis. We tend to do it laparoscopically. So this is a left-sided resection, a rectal cancer. We're seeing here the descending colon, and we're putting the stapler across in that green zone. So, I mean, we're pretty good generally at making that decision as to where best to transect. But this is the bit that's going to be part of the future colorectal anastomosis. So it's really very important that that it's well perfused at the time. We'll also give the ICG again after we've made the anastomosis. But the point of giving it now is that if there is any impairment in the blood supply, you're able to see it and do something before you go to the effort of constructing an anastomosis. And generally what you do is you move more proximally into a better perfused part of bowel. Now, I mentioned we're pretty good at deciding where to go, but sometimes this technology changes our view, sometimes by just maybe a centimetre to see here. Or in this video, you'll see actually where I thought I was going to make my transection was wrong by maybe 20 centimetres or so. So it seems from most of the clinical experiences, about 5% of the time, giving this drug routinely will give you some information that will make you change where you're going to make your transection point from. And that seems to then translate into a reduction in anastomotic healing complications. Here's it just been given at the time of the anastomosis being formed. We've already given it earlier at the time of proximal transection, but we're just confirming that in situ, just before we close the stapler together, that this is indeed a tension-free, well-perfused anastomosis. So this technology has been around for maybe about 15 years, and quite a lot of people have been doing quite a lot of work on it. We ourselves did work 5 years ago now. This is a phase 2 trial, so this isn't proving it works, but this is looking at an association between outcomes and use of this. 3 centres, Dublin, Geneva and Oxford. And this is just a comparison of outcomes with regards to anastomotic leaks in colorectal operations when patients had been given ICG in comparison to a control group who hadn't been given to it. And really what it shows is that the colorectal anastomosis, particularly the low colorectal anastomosis, seems to have a reduction in anastomotic leak of nearly 2/3, going from nearly 11% down to 3% in an associative fashion. Now, this isn't a causative study, it doesn't prove the mechanism, but we haven't really seen such leak reductions with specialization, with centralization, with changing from open to minimally invasive or laparoscopic to robotic. So this seems really very interesting. And there's very many studies done now on it, going from phase 1 to phase 2, and some indeed phase 3, some randomized controlled trials. They're generally been a little bit small, but what you're seeing across the literature is a very consistent benefit effect in colorectal surgery with regards to anastomotic leak rates. Now, what has me really excited at the moment about this is that there's a number of large powered randomized trials just concluding. Anyone who went to the European Association of Endoscopic Surgery might have seen June Watanabe presenting the results of the predominantly Japanese study Essential. They had over 800 patients in it and showed us a significant difference in leak rates in rectal cancers. Now that paper has been submitted for publication. I guess the devil is in the detail, but it was a very exciting presentation to give at the meeting. I'm co-PI with David Jane on a trial led out of Leeds, which is called the INTAC study. Again, powered to 800, but it's just at the interim analysis phase, having reached 550 patients. Now, this trial included a 3-month follow-up, very high quality control measures, including asymptomatic leaks. Everyone gets a gastric enema, whether they had a defunctioning stoma or not. So the interim analysis is on 550 patients, but as the trial continues to recruit, in fact, there is going to be data on 650 patients. So hopefully in January or February, we'll get a sense of what the interim analysis results are going to be. There's another trial in the Netherlands. They're very successfully recruiting. They've over 700 patients now. And then I understand there's a further randomized trial called Poster from China, but it's just hard to get any detail on how that's going. But very soon there should be very high quality evidence as to whether this works or not broadly, and how well it works or not. So I think that's potentially then practice changing depending on the results of these trials. This though is changing colors on a screen, and there's contrast being put into it, and it's quite interesting now with advances in computer vision and how you can maybe quantify flow dynamics. We're all used to surgeons making decisions based on the appearances that they see, but here we're starting to move towards computer supported quantification of perfusion possibilities. It's not really about whether the tissue gets green or not, it's about how quickly it gets green. And that inflow of dye arterially tends to happen very quickly. So having a computer then to kind of quantify that could be very helpful in bringing further information out of what might be useful. And here you see, again, the bowel being prepared for transection, but the software that we built as part of a research collaboration with IBM research between the Mater Hospital, UCD and RCSI is showing that we can quantify the data and then apply rapid statistical analysis to it. Now, once you can do that, it's really very interesting what you can make this dye do, the combination of understanding physiology in a pathological situation. And that's basically a big part of this other grant we're just nearing the end of, using this type of methodology and dye to identify and characterize cancer in tissue. And it's just important to say here that ICG doesn't have any preferential accumulation in cancer tissue. It's just a dumb dye that circulates. But we all know that cancer tends to distort the tissue it's in, and there will be a different perfusion flow in cancerous regions versus benign or normal regions, or at least that was the hypothesis we had at the outset of the study. So it's been a big, broad-ranging project. It's been terrific to have Donal as a collaborator with his expertise in dyes and chemistry. There's been an AI and computer vision component through the capable team at IBM, and there's a pathological group, a startup company called DyeCyfex that we're looking at automated analysis of pathological slides. But to give a little brief indication of how this is working, we have a rather simple user interface. You visualize the tissue of interest here. It's an endoscopic picture of an ulcerated lesion in the colon. You pick regions of interest that you're going to compare. The dye is given and circulates through all those tissues, but tends to have an impeded flow through any tissue that is cancerous. You'll see here now, as I was talking, the dye has been given, it's been circulating. You'll see that black and white screen become white and the dye frames coming through it, so that there is altered, different flow, which can be distinctive if you're comparing at the same time a region of normal tissue with a region of abnormal tissue. But rather than asking surgeons to interpret graphs now instead of pictures, the computer is able to take statistical inferences of different points along that graph, and within 50 seconds come up with a decision as to what that tissue is, as to its behavior in response to that dye. This was the first cohort study done. This is the training set, and we really were getting very high accuracy both at individual regions of interest and at whole patient. And we've improved that now up to 85 patients, and it's holding out. And we're moving forward now to a bigger study of 500 patients with 5 leading cancer centres. But that's just with this rather dumb dye being used to it. Donal, you're going to tell us about intelligent dyes and personalised dyes. Great if you might come in and help me out here with the really smart stuff.
Thanks very much, Ronan. Let me just share my screen.
Stop presenting and give over to you.
Thanks. Let's try that again.
Yeah,
That's
we
good.
see you.
Okay. Thank
Yeah.
you so much. Thanks, Ronan, and thanks, President, for the opportunity to speak today. What I'm briefly going to try and show you from the research in the Chemistry Department at RCSI and in collaboration with Ronan and IBM researchers, where we're trying to bring these concepts in terms of using fluorescence in surgery a bit further than what ICG can deliver for you. So you've seen obviously there from Ronan what ICG is used currently clinically for and how we're trying to develop it further, but it has some limitations to go beyond those types of applications, and they pretty much are determined by its pharmacokinetics, because as Ronan said earlier, it's exclusively excreted through the bile, and it has an extremely short half-life, about 4.5 minutes. And this actually helps with some of the applications that it's currently being used for, but it hinders its further development because it has pretty much passive localization in vivo, and that basically means that you can't direct it to any specific tissues. So this has led to a wealth of research development in our own team, but also in other international groups. And some of the types of things that people want to bring this type of technology to is to develop real-time intraoperative imaging for preservation of vital structures. And one example might be the ureter, I'll show you in a moment, for the detection of cancer metastasis in lymph nodes, so that you can detect these during surgery rather than post-surgery. And also then the one that most people really want to try to achieve is a targeted tumor margin delineation, so that you can essentially guide resection in real time during the surgery. Unfortunately, that might be challenging with indocyanine green, so we have started from the beginning. We develop our own fluorophores where we can control the properties of them so that we can tailor them essentially to what might be surgically of use. So this is the general structure, which we don't have to dwell on, other than it can be manipulated, and not only can it be manipulated to target specific tissues, it can also be manipulated for its color. So this is just to give you a sense of how you can think to the future, that in not just having one color of indocyanine green, using a different fluorophore with a different color simultaneously, you can see both of them in different regions at the same time. So this is just two vials with ICG and with our fluorophore. But in essence, by just changing the wavelengths that we're detecting, you can see these two fluorophores simultaneously. And there's clinical machines emerging now with this type of technology. So this has the potential for essentially tissue color coding, which again could guide more than one tissue or one part of the section at different times during the surgery. So our goal is not necessarily to replace indocyanine green, but just to develop new applications that it is not possible to be achieved with indocyanine green. So to give you a sense of how that looks, here we're looking at imaging of the urinary pathways, essentially using the core fluorophore, but decorating in a chemical fashion so that its excretion is rapidly through the kidneys. We can, within 10 minutes, begin to visualize the ureter. And this is in a porcine model carried out in the Institute of Image-Guided Surgery in Strasbourg. And with IBM, we would develop some nice software to demonstrate that. So what you're seeing here at the top is a heat map of fluorescence and the pulse of urine going through this particular ureter in real time. Below, you can have the white light view and also the fluorescence overlaid here in white. And with this particular agent, it means we can identify the ureter within 10 minutes post its administration, allow its identification and its demarking, and hopefully preserve it during the surgery. And it can still remain visible, the ureter, up to 60 minutes. So it's not a very short window, it's plenty of time to identify it and to mark it out. Another example of where we develop new probes is for lymph node mapping. And this is, again, another porcine model using the stomach wall model, where we're just introducing a localized injection of the fluorophore. Again, looking at our heat map at the top here, this big here, red mark here is in fact the fluorescence coming from the syringe as it's been injected into the tissue. And when it's removed, you'll see then the local site of injection. Again, below we have the superimposed white light and fluorescence, but perhaps it's more easier to see it in this new way, looking at just a near infrared fluorescence intensity heat map. So following a quick massage of the site of injection, you can almost immediately begin to see here new fluorescence emerging. That essentially is the nearest lymph node to this site of injection. And you can see these lines on the image. Essentially, they're the lymph node channels or the lymphatic systems going to these particular lymph nodes. And this is unedited video, so within a matter of moments, you're beginning to see the lymph nodes connected to this specific tissue. And Pause just for a minute there. There's another one appearing here. After approximately 5-6 minutes, then all the lymph nodes connected to this site of injection will essentially be fluorescent and can be mapped out. And then the final one, which I just want to briefly show you, is our approaches towards tumor margin delineation. This is the one that many groups are really working quite hard on. But there is a significant challenge with it, is that even when you have a good fluorophore, and you may attach some targeting agent to it to direct it towards a cancerous tumor, there is a problem, is that you always have fluorescence from the fluorophore as soon as it's administered. So if you can imagine the administration of a targeted fluorophore, and we want to image this region here immediately after and for prolonged time periods after introduction of this fluorophore, it's very difficult to get good contrast for the region of interest, which might be our tumor versus background. And what is happening now in current clinical trials is that you have to wait for prolonged time periods for clearance of the fluorophore from normal tissue and retention in the cancerous tissue to essentially be able to get a good image of the tumor. And this is now taking days and sometimes out to weeks to achieve this discrimination, which of course is not going to fit into a surgical workflow or become practical. So we describe this quite often as waiting for nighttime to come to see the moon in the sky, even though the moon may be out in the middle of the day, but you don't have the contrast against the bright sky to see it. So what we've been doing is trying to make smart fluorophores that essentially are administered in an off state or switched off, but they turn themselves on in some form of a biological response to a local environment. So what we're thinking about here is that we would administer the fluorophore targeted in an off state, but that it would first of all accumulate in this region of interest, which would be the tumor, and turn on there first, and we can capture that image at an earlier time point, pretty soon after the actual injection of the fluorophore, and allow, with the, using image capture, collect the dynamics of that and essentially get a tumor margin identification. For that, we need a fluorophore to turn on first in our region of interest, our tumor, and then we're looking at doing real-time dynamic imaging in that area, akin to some of the type of software technologies that one was showing you a moment ago. Essentially, though, what we're trying to do from a chemical engineering point of view is we need to develop our molecular light bulb that's able to switch itself on in the right place and at the right time. So just to give you an example of how that can be done is that looking at this particular fluorophore, which is a responsive one, it's called a pH responsive one, and that means it's at a physiological pH of 7. It is not fluorescent, but upon uptake into cancer cells and trafficking into acidic vesicles, it will switch itself on. But we still have to bias the uptake into the cancer cells, so we use this particular peptide sequence. Is called a cyclic RGD peptide, and it's excellent at recognizing integrins that are overexpressed on cancer cell surfaces. And just to give you a sense of how that impacts on the imaging, a very similar structural compound here, the same targeting agent, but we just changed this responsive off to unresponsive component. We put in a benign solubilizing group. So this one here is always switched on, essentially, always fluorescent, whereas this one switches its fluorescence from off to on in response to cellular uptake. When we look at this in small animal models, using subacute tumors, this is an MDA-MB231 subacute tumor or human breast cancer cell tumor. Looking first of all at the peptide targeted always on fluorophore, if we administer this to the animals, we get what we would anticipate is a highly fluorescent animal immediately, because the fluorophore is throughout the body and we can see no discrimination for the tumor. In contrast, with fluorophores that switch from off to on, at earlier time points, within 1 hour, hopefully you can see here in the picture here, this is the tumor. The tumor is beginning to accumulate the fluorophore, and because there is very little other background, bar this small background here from the liver, we can essentially begin to see the tumor. In the traditional fashion, if you have a fluorophore that's conjugated, that's accumulating and retaining, you have to find the right time point where you get the best contrast between the tumor here and the background, which might be somewhere between 6 hours roughly. Or you can wait a very long time till it's fully cleared, and then the remains of it are still within the tumor here at 48 hours. But in contrast, when it switches from off to on, in this time point from 1 to 3 hours, we actually get the maximum differential between tumor and background. That allows us then potentially think about how this could be done intraoperatively. So when we look at that uptake into the tumor and how it impacts the fluorescence in this early time point following administration, we see that within 120 minutes, when we graph tumor intensity of fluorescence versus time, we reach a maximum. So with a maximum emission being reached within 2 hours, there is the potential for agents like this to be administered intraoperatively, and then the images collected at an early time point of the surgery, that image is being processed, and then a tumor margin being identified before the resection commences. And just from our animal study, we see quite gratifyingly that obviously when the tumors are resected, they are quite fluorescent. And when they're dissected, we see that it is in fact the margin or the outer periphery of the tumor that is brightest in this type of approach. So from our clinical goals, and we look into the future, what we're looking to try to develop are real-time intraoperative tumor margin delineations. And as I alluded to earlier in the talk, the detection of cancer metastasis, because what we would think about is if upon fluorophore administration, if we can identify the tumor margin, but also if we send it through the lymphatic system where it is switched off, but wherever it detects cancerous metastasis in the lymph nodes, it will switch itself on, and that might be able to lead to the identification of these sites during the operation rather than post-surgery. So that's just a quick snapshot of what we're trying to develop in collaboration with Ronan and with IBM, and I'm happy to take any questions.
Donal, that's really fab. Thank you very much. It's amazing what you can make visible or easier to see. So this is brilliant science. You have it all working in animal models. What are the sort of the next steps now to proving that in humans, in human operations?
So I guess it's the age-old challenge of how to clinically translate it and to find the right pathways to do that, whether we go straight into trying to do a clinical translation for the cancerous, or we start looking at more easier structures such as the preservations with ureters. So what we're trying to do is do a dual track approach to be able to achieve both ultimately, but just trying to progress them step by step with clinical trial centers.
Yeah, so Simon, you're still here. I was going to be cheeky maybe and ask you a thought. You know, I mean, like what Donal's exploiting is the phenotypic environments that are, of course, being created by the genomic patterns underlying the disease. Can both of you sort of imagine maybe us seeing genomic behaviors more directly? Can you kind of put on your science fiction hat and see that could be something seen in operation?
Well, even just looking at the mouse models and incorporating the single cell omics into that might be really exciting to do. I'm not sure exactly what's possible, like in terms of patients, but if we start off with those mouse models, that would certainly be something that would be exciting to do.
What exactly? What would Donal? You're smiling too. Do you think do you see synergy here?
Absolutely, yes. With some of Simon's stuff in obtaining tissues, obtaining markers, obtaining different differentials that could be exploited. Whether we can get the speed of the genomics profiling quick enough to satisfy a surgical team, I'm not sure, but yeah, it would be interesting to see. It's definitely getting quicker and faster.
I guess we could, with what I was alluding to with the single cell approaches, I guess we can look at differences, like in terms of which regions are maybe responding better or coming up first, and the markers behind those, and potentially how that might translate to the human situation.
And Donal, I mean, you've been talking about kind of software to kind of improve the accuracy and the speed at which we recognize these type of differences. Linking the software then to a therapeutic mechanism, are we back to photodynamic therapy or something like that, or do you think this brings us into robots?
I think both, actually. I think the software component is very, very important. Even what, you know, visually as I show those images, these are complex images, and to make on-the-spot decisions on those images alone is going to need additional supports, no doubt about it. But when you get acceptance of those supports, you really can see the power of them. Also, then obviously, then where you can surgically resect these agents can be turned into photodynamic agents that would clear the margins of tumors as well. That's another possibility, and that's technology we have developed but not managed to clinically translate it yet. So that's another possibility, yeah.
Yeah, I mean, I think it's awesome. Those watching at home, please do text in or give your comments. I think robots is a part of surgery now. I'm going to link that on now to Colin Pearce, who's in Limerick. Colin is a general and colorectal surgeon. Colin, you've been using a single robotic platform for some time now with good outcomes in Limerick. We're looking forward to your talk, and particularly, I guess you're going to tell us or educate us on the different type of platforms and making these systems smarter so they can actually do something that we can't currently do, and maybe bring in some of the concepts that Donald and Simon have been saying. How do you think that's going to look, and how is RCSI going to be part of this sort of new era of augmented surgical support? So Colin, over to you.
Thank you very much, Ronan. Thank you, Mrs. President, also for your introduction, and of course to RCSI for inviting me to contribute this evening. And I'll just keep this nice and simple because while there may be a huge number of robots and very fancy things happening, we have to be realistic as to what we are going to see in our hospitals for both our use and that of our trainees over the upcoming years. Of course, it's important to go back to the beginning and the dawn of the robot. And here we see the Puma 560, the first surgical robot per se, used to perform a neurosurgical biopsy as far back as 1985. So 37 years robots have been in use. But now, of course, what we're familiar certainly here on Irish soil is with this, the Da Vinci robotic platform from Intuitive in Sunnyvale, California. And here we see their latest version, or second latest, I guess, the XI, with the actual robot per se on the left-hand side there with its 4 arms. And then we have the stack with the vision stack and cart in the theatre on the right-hand side. And this is just a quick look as to how the robot, for those of you who haven't been in a robotic theatre or don't use robotics, this is it with the 4 arms with instruments via ports into the patient's abdomen in this case for a colorectal operation, and then the surgeon sitting at a console here on the left. That's just part of our audiovisual team. We record all our operations for both training and learning. And then you can see the movements of the surgeon's hands are mirrored at the bedside by the movement of the robotic arms. And we have both coagulation and cautery available across all instruments. And I guess really what makes the robot even unique is that the surgeon is in control of everything, camera and 3 operating instruments, versus in standard keyhole or laparoscopy, you're reliant on someone holding the camera for you and indeed possibly utilizing an instrument to assist you also. We have a bedside assistant who does utilize a further instrument. So really we have 4 assistant or 4 operating arms in play. Robotics started initially in the private sector in Ireland with our urology colleagues, then in the maternity hospital in Cork with our gynecology colleagues, and then we established a general and colorectal program here in Limerick, and since then the sites have really started to grow, and my understanding now is that we have 12 platforms countrywide. And indeed, for my speciality, colorectal surgery, there are 8 rectal cancer centres, and there are robotic platforms in use in 7 of those. And my understanding is that the 8th in Waterford is actively looking to get a robot in, and hopefully that will be sooner rather than later. The robot itself has progressed over time, and there have been a number of iterations of the Da Vinci. The latest really in mainstream use now, 2017, the Da Vinci XI with the X and SI and various other ones prior to that. And of course, it's nice to do operations with the robot, but can it help us in any other way? And two operations that I find it very useful for are these, transanal minimally invasive surgery, or TAMIS, on the left, and a right hemicolectomy with an intracorporeal anastomosis on the right. In terms of the TAMIS, of course, this has been performed laparoscopically for quite some time, but as you will see toward the end of this operation, you have quite a large defect in a narrow confined space that requires suturing to close it. And unless you're very skilled at laparoscopic suturing and doing it on a regular basis, that will prove tedious, if not indeed impossible for a number of us, and will take an awful long time to do. Suturing with the robot is, as the company name tells us, intuitive, and closing these defects is far, far easier. On the right side, while we don't really know of too many oncological benefits to date for using robotic platforms in the colorectal context, there are certainly a number of functional benefits. And when we perform an intracorporeal anastomosis, as you see here, we've certainly shown locally in Limerick, and that would be mirrored by my colleague Sean Martin and his group in St. Vincent's, that these patients go home at least a day, if not 2 days sooner. And in saying that we would discharge these patients on day 3 or day 4 post-operatively, all going according to plan, which is quite reasonable for a oncological right hemicolectomy. And again, the suturing obviously speeded up here is far easier. You can stitch forehand, backhand, left hand, right hand, all very interchangeable and very easy to do with the robotic platform. Complete avoidance of bowel handling, virtually no episodes of ileus in this cohort, very little post-operative pain, reduced incisional hernia incidence, and most notably in an Irish healthcare context, early discharge and freeing up that hospital bed. Other things that the current robot will do for us, well, the Tile Pro function, as seen here with my colleague Professor Sebastis Giri, one of our urologists, where you can have real-time ultrasound by plugging in simply to the back of the robotic console, and then, as you see here, what he's looking at on top is the actual inside of the patient's abdomen and the kidney and the tumour he is going to take out, and is able to see real-time ultrasound where that is, as opposed to traditionally where you might need a formal ultrasound machine. So this is very useful for our urology colleagues, hepatobiliary, not in such wide use in colorectal surgery at present, but I suspect that that will change as time goes forward. But a very useful adjunct. And indeed, to put the colorectal hat on, our colleagues in St. Mark's in London, Danilo Miskovic and his team use this in a very unique way, where here we can see they take individual patient scans in terms of the vascular anatomy. Not something that maybe we pay enough attention to as colorectal surgeons. On a pre-operative basis, we're interested where the tumour is it involving adjacent structures. This is for right colon cancers in terms of ureters, duodenum, etc. But we don't maybe look in depth at the vascular supplies. So here they can map that out with an individual patient's CT scan, create a 3D model, as you can see here, and know the exact anatomy of the patient they are going to operate on in due course. So whereas we often might potentially get into bleeding and say, oh, that's aberrant anatomy or this or that, that can't be the case here. You know exactly what you're going to find with this 3D model technique. And then by plugging, in this case, having the imaging on an iPad, plugging that into the back of the robot, the operating surgeon can see the patient and what he's operating on here above, while an assistant at the bedside can manipulate the 3D reconstruction of that patient's anatomy so that there aren't any vascular surprises. And this is particularly useful in what we call CME, or complete mesocolic excision. We could talk about that all day another time, but certainly I suspect that this will become something that is going to be par for the course in colorectal resectional surgery going forward. But of course, having had a monopoly on the market for so long, there are now competitors there. And probably the main one at present is from CMR, or Cambridge Medical Robotics, and their robotic platform is called Versius. They've been over, as we see here, to visit us in Limerick on a couple of occasions and are very keen to get a foothold in the Irish market. What makes them different is that this is modular. Each of the arms can be wheeled very readily from one theatre to another, almost like a suitcase in an airport, and also introducing the arms into the patient can be done via standard laparoscopic ports. Also, their business model is somewhat different. There is a very large financial outlay to get a Da Vinci robot into your hospital, whereas CMR will offer a slightly different business case in terms of that you would pay up front for the proposed number of cases you might do in a 6-month or a 12-month period. Plenty of literature on this platform now with very good results, and can certainly do all the operations that the Da Vinci can do. Probably is finding a niche maybe more so in intermediate and less complex operations than we might use the Da Vinci for. But certainly probably number 2 out there at present. Of course, when CMR produced this, Da Vinci weren't too long in launching their Da Vinci single port. Now this looks extremely attractive as an instrument. I think this would be extremely useful for performing the aforementioned TAMIS surgery, oral surgery, probably lung surgery, possibly even vascular surgery. But as we know, single port laparoscopy was all the rage maybe 15-20 years ago, and there were very few, if any, people really practicing that on a regular basis because it doesn't give you any advantage over standard laparoscopic approaches. And that's likely the case with this, albeit possibly for small orifice surgery as seen here in this model, this could prove very useful. Unfortunately, it would require you to purchase a completely new platform, but certainly the dexterity, the maneuverability in very small spaces that it can afford you is very impressive. But it will set you back over a 7-figure sum. I believe it's in mainstream use in the Far East and also in the United States. Do I see any hospitals in Ireland acquiring this in due course? Probably not, unless some very significant benefits of using this platform over another are produced, which would seem unlikely, but certainly a very fancy piece of equipment which you would be very proud to have in your theatre if you could afford same. Medtronic have joined the party and indeed had a tour of Ireland this year showing off this platform, what they term Hugo. They have gone more for the CMR sort of platform, an open console versus the closed console we see with the Da Vinci, and then maneuverability in terms of each arm can be wheeled around. You can use 1, 2, 3, 4, however many you wish, allowing this to be easily transportable between theatres up and down your theatre corridor. Not in place yet in a hospital in Ireland or the UK, as far as I know, and I'm not exactly sure of their cost structure and business case, but I'm sure there will be quite a push on this over the next 12 months and going forward. And Johnson & Johnson have their robotic platform termed Ottava, and this comes with its own table whereby the arms, as you can see here, the diagram on the right, latch on to the table. I don't know of anyone who has seen this in person yet, but we look forward to that progressing in due course. But really at present it's probably a 2-horse race. It's the Da Vinci platform from Intuitive or the Versius platform from CMR. All platforms in Ireland currently are the former. So what about the future direction? And people might think of things like artificial intelligence and robotics, etc. That is very futuristic in my view. What I hear about and see all the time is our trainees and what they are doing. And not just in colorectal, but obviously in urology, gynecology, hepatobiliary. And more our trainees are going on fellowships that have a robotic influence, if indeed not a dedicated robotic fellowship. And as we all know, and as we all heard as trainees, trainees are the future. But how are we training them for the future at present in Ireland in terms of robotic surgery? Well, we're doing it on a very ad hoc basis is the answer, but the college have noticed this, and from July onwards will be incorporating robotics into formal surgical training. Clearly, as a group of surgeons and led by the college, we will need to develop national guidelines in this context, a designated curriculum, and governance which to date has been done on a kind of individual hospital basis. Programs have been set up on an individual hospital basis, and we really need to all gather together and make this a much more formalised process in terms of robotics going forward. And then how might we train that? Well, actually, as things stand, we're probably in a very good position to train robotic surgery, probably a lot stronger than we were with the advent of laparoscopic surgery. And I'll explain that shortly. And there would clearly probably be two parts, what we might term core robotic training, which would comprise of some online learning that can be done anywhere, anytime, by anyone. Robotic platform training, so actually working with the robot, learning how to move the arms, dock the robot, how to deal with an emergency undocking, maneuverability of the robot, all these things. And that's where we have a strength. That does not need to be delivered just at the RCSI campus in Dublin, as has often been the case in terms of training days. With 12 platforms around the country, we really have a good opportunity there. Simulation, more and more a bigger part in training, and the robot, in fact all platforms, not just the Da Vinci, have very good simulator functions. And then higher robotic training would obviously be those trainees who want to use robotics in their consultancy, etc. We will be able to pick those out hopefully from an earlier stage and then get them into focused jobs with high volume robotic exposure. Cadaveric operating is obviously extremely useful, not available on all sites and probably not available on many sites in Ireland, but that could certainly be incorporated also if there are platforms in those training centres that can avail of same. And as I've said, this sort of hub and spoke model lends itself very well for robotic training. If we just take the 8 rectal cancer centres or 8 cancer centres, 7 of which now have a robotic platform, all 8 will do shortly, we hope, well then the training does not need to be centralised and indeed can be regionalised, something that we're looking at in terms of training with the RCSI, or RCSI are looking at, should I say. And thus the trainees won't have to travel up and down. There'll be platforms closer to them with a lot more availability, and these platforms can be used from a simulation perspective, etc., 24 hours a day, 7 days a week. So they're available there and possibly a lot more so than some of the other simulation and training that we have gone through and our trainees are currently going through. And this has begun, and only as recently as the end of October, in conjunction with Intuitive, there were robotic platforms in RCSI, and we had some initial training with our more senior trainees in that regard. Very well received, and we certainly look forward to this growing and becoming much more standard of training as time goes by. Finally, I would say to anyone who has a platform in their hospital or is getting one or is looking to upgrade or get new instrumentation, the ability to train is unique with the dual console. Akin to a pilot and a co-pilot, Whereas traditionally doing a laparoscopic operation, you might start it as the trainee with your trainer holding the laparoscope. You might run into a bit of difficulty. The trainer will say, just give me the instruments there for a minute, I'll give it back to you, and you never get the operation back, and there's elbows going on and you're getting pushed around. That doesn't happen with the dual console system. With the push of a button, you can give just the camera or one instrument, two instruments, or all instruments over to your trainee. You can mark out exactly where you want them to dissect, and nobody's getting in the way of each other, and the operation can be transferred between trainer and trainee, or indeed between trainer and trainer, where you're doing dual consultant operating in, say, a complex pelvic case. And it really, really is unique. And we have shown with Jarlath Bulger in one of our earlier papers from our group that yes, this will make the operative time at the console that little bit longer, but with no difference in terms of outcomes. So a very, very unique and excellent way to train. So I don't have too much more to say. Once again, thank you very much for inviting me along. I hope that's given you a bit of a flavour where we really are at the moment in terms of robotic platforms around the country, some of the advantages of it, the clear advantage of how we can train on it, and how we will go about training in conjunction with leadership from the RCSI and incorporating robotic surgery into the curriculum going forward. Be happy to take any questions, and thanks again for having me.
Kevin Barry's in the audience, so the president suggested maybe Kevin could come in and just give the college's perspective, and then after that it's 7:10, and in the interest of everybody's time we might wrap up, and we'll send a video of the full webinar to everybody in the morning, if that's okay. So maybe Prof. Barry, maybe if you could come in and give the college's perspective of, you know, where you would see training going and how RCSI are going to support that going into the future.
Thank you, Pádraig, and thank you indeed, Colin, and indeed many of the points that Colin has made this evening. We would agree with the philosophy that you've outlined, Colin. RCSI has definite plans in place to review or to introduce formal robotic surgical training. We've had consultations with Professor Tony Costello. Those of you who are not familiar with Professor Tony Costello, he gave the international lecture at the Frere Symposium in Galway in early September, and he spoke about robotic surgical training. So we've had a number of further meetings and discussions with Professor Costello, who's based in Australia. About 20 years ago, he set up the Australian Medical Robotics Academy, abbreviated AMRA, and he advocates a curriculum, if you like, which we're quite attracted to, and it contains 4 steps, if you like, which we feel that trainees can go through in terms of robotic surgical training. And as Colin outlined, the steps would include what you might expect, namely exposure to initial theory and educational self-assessment, then followed by training in terms of simulation, and then moving on from that step onto step 3, which would be exposure to training in a dry laboratory type environment where you would work on benchmark models, and then eventually, once you've built up enough skills, to the live operating theatre. And as Colin mentioned, there are plans to modify the RCSI logbook, and indeed the ISCP will have to do this as well, to capture robotic surgical training for all of our trainees. The philosophy at RCSI would be that robotic surgical training should be introduced at the very start of surgical training. So what that means in practical terms is that in July of 2023, we would envisage that robotic surgical training should be up and running, including first-year core surgical trainees, and that we will probably adopt most of the principles that Professor Costello has developed in the Australian Academy. So there are plans indeed to introduce a curriculum. And as Colin outlined, obviously it can't all take place in one location. So there will be, if you like, a hub and spoke model, just like you said, Colin. York Street will have access to the robot for trainees to come into the college to practice their simulation skills. But I agree that it will be necessary to use all of the centres in a coordinated fashion across the country to facilitate robotic surgical training. And we would advocate that robotic surgical training should be available throughout all of the 3 phases of surgical training, extending from core training into higher surgical training. So certainly most of what you said, Colin, I would agree with. And we have plans in place to introduce a formal training in July 23. And indeed, our president, Professor Laura Vianney, is heading out to Australia in May of next year to meet with Professor Costello to, if you like, solidify these plans. And I don't know whether Professor Vianney wants to add any further comments to that.
No, I just suffice to thank you all very much for presenting this evening, and it's giving us a glimpse into the technology shaping the future of surgery, and it's something we're all going to have to embrace. Well, not personally, I will have to embrace at this stage of my life, but certainly all the young people will have to embrace. So I think it's quite late now, so probably we won't take any questions at this time. But just to thank you all very much, and to thank the Surgical Affairs, Mr. Porrick Kelly, and to thank Janelle, and to thank Catherine Jordan from fellows and members for helping us this evening. Now, we have missed some of the recordings this evening, so what we'll do is we will get a recording of the one part that we've missed, and we'll send it on to anybody who would like to have this. So let us know, we'd be delighted to pass it on to you. So we are all facing the future, and at Charter Day we will be the sort of the theme of the Charter Day is shaping the future. So we hope we will see you all then. Maybe we can take this a step further, the thoughts and ideas of this evening. So again, thank you all very much for listening in this evening, and good evening to you all.